So you were told that for certain cancers the metastasis will happen and they'll go to a particular organ first. Like there's a tendency to go to first. So what governs that? Is there something specific?
I don't know that part, but I am assuming कि जैसे different kinds of cancers होते हैं ना, उनको different kind of environment, जैसे हम लोग हैं, हम लोग को कुछ environment is better than the other environment. So breast cancer metastasizes preferably in the bone, for example. So bone environment is more conducive to their growth. It could be also a proximity issue. Liver cancer generally lungs में जल्दी जाता है. But it could also be that lung atmosphere is more conducive for that liver cancer. So like I was telling you initially when I started, I was an organic chemist, right? So I had no knowledge about biochemistry, glycolysis, nothing. So I used to naively think cancer is cancer. One cancer is same to the other cancer. That is the most naive thing to think that. So every cancer is different. Every cancer responds differently. Every cancer. So they like different kind of external signals. So that is why they tend to. But having said that, many cancers, they metastasize to the bone.
Yes, ma'am. Another similar like attached doubt. If there is metastasis happens, so from the organ of origin, the cell will have a particular structure that is suited to that organ. So that cell will metastasize to some other location. So what would be the changes? Like if it goes from, let's say, breast cancer to bone cancer, to what will there be significant morphological change, or is it like a intermediate state where it works in both places and it's adaptable?
So one thing about cancer is that, remember when I was telling you TNM staging yesterday, right? Tumor nodes and this thing staging. So one thing they look in cancer is that how differentiated it is. So suppose hepatic cells look like this, right? Hypothetically. So cancer, when it starts, it will look like this, and then it will look like this, then it will look like this, and then it will become more round. So it will lose the shape of that thing. So this is the differentiated stage, right? So from differentiated, it becomes less differentiated. It has changed its morphology. Now they are more round on top of each other. So when the pathologists look at some cancer to tell how aggressive it is, if it is differentiated, it's good news because it is still looking like normal cell. The less differentiated it is, the more aggressive is the cancer. So by the time it metastasizes, it's no longer that original thing. It's completely different structure. So this will go and this will metastasize somewhere else, right? So that is one aspect. Because this cancer, this metastatic, these cells no longer look like the parental cell. Okay, and when they go to the bone, they don't have to look like bone cells or that because it's so metastatic. So that is one part, and that is universally true across the board. But having said that, remember I told you about EMT today, epithelial to mesenchymal transition. So one of the ways cells metastasize to other organs is that they have this epithelial morphology, right? And then they become completely mesenchymal like this, and they squeeze through hole. So this is called epithelial to mesenchymal transition, right? And they go somewhere else and lodge. Then there is a reverse process which answers the questions. Mesenchymal to epithelial transition. So they become more like that. Now they will differentiate. Then they will form more of those kind of cells, right? They will put their anchor down and grow a whole colony. Then some of them will become mesenchymal again, and then they will go to another thing. But they don't have any pressure to look like bone cells or anything, because they are not trying to merge with that. They are so aggressive, they can be on their own.
One more small doubt. Does the does the differentiation within a tumor cell show some sort of, is it indicating towards the division of labor possibly, that they have separate functionalities and they all act together in some sense? Or is it just morphology randomly happening?
No, their morphology is different because, so when a cell is, so let me go back few steps here. Why do cells differentiate to start with, right? So when there is a zygote, right? So it starts with a single cell and then it divides and divides and divides, and then the cells, there is a division of labor, the cells differentiate and different organs are formed and everything. Cancer cells are not doing liver function, right? They don't do anything that the liver cells do. Their role is to just proliferate. They are not contributing to our physiology or anything, right? So they don't have to differentiate at all to do any function. What function they are doing in our body? Nothing. They are invaders, right?
I was talking in a sense that they are cooperatively working towards proliferating better, I guess. I think that's linked metabolism in some sorts, like if they are doing something like that.
Cancer cells, when they help each other, that is the example I showed yesterday, right? The hypoxic cells are giving lactic acid. So if they are working together, they are only working among themselves to survive. They have nothing to do with the organ.
So it's not a specific process they are doing, it's just transmission of substances they are doing. That's why they don't need specific structures like...
Yeah, and their abnormal structures allow them to escape from their abnormal structures allows them to squeeze through spaces to invade that. So they don't do anything for the good of us. They are just invaders. Yeah, they are just bad. There is nothing good about cancer. There's not even a single thing good about cancer. Our body is not getting benefited in any way. At least we don't know that in any way they steal stuff. They will make these long tubes, steal stuff from normal cells. Yeah. And then you know about exosomes. They will release these small vesicles filled with, all the bad stuff, and they will go and invade neighboring cells, unsuspected cells, and then they will make them carcinogenic. So it is all, they contaminate the neighboring cells also. So it's really all bad things, yeah. And they are so powerful that they don't have to differentiate to hide themselves. But then they are also, it's all like they express these receptors, right, to quieten things. So they will have all these fake receptors to fool the immune system. So their whole energy goes in protecting themselves and proliferating. Yeah.
Ma'am, I have one question. Ma'am, we talked like in case of synthetic lethality when BRCA mutation is there, so we have one option of like using inhibitors, but ma'am, PARP enzyme has different isoforms and there comes a question of isoform selectivity. So do we have a chance like delivering BRCA, like functional copies of BRCA in case, like thinking about gene therapy instead of using inhibition approach?
So the thing is that it is much easier so synthetic lethality is relatively a new concept, right? So if synthetic lethality works, you can have a very quick response like with PARP inhibitors, right? And a huge percentage of cancers are BRCA1, BRCA2 deficient. So when you develop a drug like olaparib, right, you can just immediately commercialize it, and worldwide it helps. There are so many tumors which have BRCA1, BRCA2 mutations, right? So that is very easy. You definitely can do gene therapy, but gene therapy is very complex process because you have to take out the cells from the same patient, then you have to engineer it, then you have to put it back in the patient, right? So it is so expensive, and it may take very long time to do the whole engineering part and put it back. And then you have to also think, I don't know how many times you have to do it, like probably one time adding the gene is not sufficient, right? So if you can make a drug which is orally available, or even if it is given intravenously, that is preferable because it is more cost effective. But gene therapy, people are working towards gene therapy, and someday it will become more and more accessible as we learn about it. But gene therapy is definitely possible. So if you activate P53, or if you give back BRCA1, BRCA2, one day it will become very easy. People don't have to undergo all of these operations to remove the ovary or remove the breast. They can just reconstitute BRCA1, BRCA2, and it is going to happen one day. I am also very interested in muscular dystrophy because I know somebody who has it, right? In muscular dystrophy, you have to do gene therapy. So the muscular dystrophy is a really devastating disease. The person cannot get up from the bed. It happens to younger people. So imagine your whole teenage, 20s, 30s, you just stay on bed because you don't have strength to get up, right? So that's very sad. Like the time when you're supposed to have maximum energy, you can't even lift your hand to do something. So, but relatively small percentage of people get muscular dystrophy. So companies are not interested in investing a lot of money in it because they don't make so much profit. So the therapy trials are still going on. And one day it will be realized, but the thing is, it is going to be super expensive. So how many people can afford that kind of thing? Like millions of dollars, right? So millions of dollars from your own pocket, how many people can afford that kind of therapy, right? And you have to have world-class facilities for, suppose somebody has to take out your own cells to engineer, right? But trust me, it is going to become a reality and it is going to become cost effective eventually, right? People are going to be able to afford it, and this will become true someday. So right now, even olaparib discovery that it works was a big help in this field. But you definitely can do it, and maybe 10 years from now or 20 years from now, it will become a routine process. See, you guys know that people used to have, like people used to die in 40s and 50s before, right? In my own lifetime, I have seen median age increasing. So if you can see that, so you can imagine that it is going to improve more. But we are also going to create more environmental pollutants, right? Like Venkatesh was saying, these things were not there. So we create, then we find a drug for that, our own creation, right? This is the drama, right, which will be there. Go ahead. Yes.
And there are several classes of like DNMTs and HDACs available. So like I assume in cancer and other diseases there will be certain HDAC or certain DNMTs that will be like higher in expression.
That's correct.
So my question is that like whatever is available in the market, like the inhibitors, HDAC inhibitors or DNMT inhibitors, they are all global inhibition, like global HDAC inhibition and global DNMT inhibition.
That would be bad also, right?
Yes, and also it is reported that in our gut microbiome, the microbiota produces short chain fatty acid, which also acts as a HDAC inhibitor.
Yeah, yeah.
Like, why is that so? It is very difficult to target specific HDAC or specific DNMTs. So all those molecules reported, these are all global inhibitors, not specific. So why is that? It is because of structural similarity?
That is because of the structural similarity. So, I don't work on HDACs, so I don't know much about the pockets of HDACs. But what happens among family members, that they have very, very similar pockets. So, suppose you take any enzymatic class, right? Any enzymatic class where there is a super family of proteins. What is super family? You have many, many, many members of that. And if they all use the same substrate, their pockets are going to be very similar to each other. So when you make an inhibitor, there is a likelihood of cross-reactivity. So, for example, I work on kinases, right? And all kinases, they bind ATP. So all their pockets look very similar. So there is a big joke among the scientists in the kinase field. So when Gleevec was developed by Novartis, so when a company works on a drug, so when you guys work on something and you publish a landmark paper, So how many people are involved? Like maximum 10 people from a lab, right? Maximum 10 people, right? So 10 people are there. Usually there are 4 or 5 authors and one is the lead author, and then you have your advisor, right? But when a company is developing a drug, there are hundreds of people which come. So there are many scientists, many people who work on molecular modeling, biologists, and this and that. So the first author is not the person who did the main work. Usually the first author is the person who is right at the very top. He or she has not done any experiment, but he is the one who is running the whole program. So these are the group of scientists at the lowest level, at this level, who have done all the actual experiments. Then their bosses, and then their bosses, and then their bosses, and then there is the absolute head of that. So they all come global, like so many people are there. So the person who was the head, who ran the whole GLIVAC program, right, his last name was Fabrio. I know him pretty well, right? He was that head of the whole program. So all those landmark papers of GLIVAC, they are Fabrio's paper. We don't know all other authors, but it is Fabrio's paper, right? So he famously said, there is no point in making kinase inhibitors. They are going to inhibit all kinases. Now he tells himself this as a joke, that that's what I thought. Because that time people thought you could never make a kinase specific inhibitor for one kinase, because they all bind ATP, the pockets are all similar to each other. But GLIVAC is specific for BCR-ABL, because now we have discovered that there are some kind of pockets inside which are specific to different kinases. So if you make an inhibitor, you can do it. Second thing is that kinases have inactive state and active state. So when they are in active state, they bind ATP. When they are in inactive state, they don't bind ATP. So what is the unique thing about that? In the active state, they bind ATP. So all the kinases in the active side, they bind ATP. So when they're in the active state, their pockets are very similar to each other. But when they're in inactive states, their pockets are actually different from each other, right? So if you make an inhibitor which binds the inactive states like GLIVAC does, you can make a specific inhibitor. So now coming back to HDAC and DNA methylases, it is possible to make specific inhibitors. But keep in mind, these are relatively new field. Epigenetic itself is a new field to start with. So we have made inhibitors, but we are going to make more and more specific inhibitors. And also with time, we will know more substrates of each of these DNA methylases and histone acetylases. So then we will know, okay, this histone acetylates these things. This one does this thing. So which one is better to target? Do you see that? Then things are not that simple. Everyone has favorite substrates, less favorite substrates. So how much we should inhibit versus how much we should inhibit this target. So you can do a lot of fine tuning also. So eventually we'll get to better specificity, selectivity.
The thing is, like in case of pocket identification, so there is a lot of computational work that can predict new pockets.
Yeah.
That is prediction. So can you just elaborate how can we confirm these can be a novel pocket that can be targeted?
So this is a very easy thing to do, surprisingly. Okay, this is relatively easy thing to do. So have you heard of AlphaFold? Right? So you can do docking with AlphaFold also, and you can predict different kinds of structures and that, and then you can modify your compound. This is something I have seen for the last 20 years. People are doing that all the time. We have done in our lab also. And we can predict new pockets, and then we modify the compound accordingly, and it really works. It really works. So suppose you identify a pocket, and you see that that pocket is a linear pocket, like narrow pocket, right? It is not like this thing, but a narrow tunnel is there, right? And your compound is there. So if it is a narrow tunnel, you want to add a simple chain here, not like an isopropyl group which is branched, right? So you make a propyl group instead of an isopropyl group, and that will be a straight, and if you can access that pocket, it will increase the affinity of that compound, right? And then you have to test it experimentally by making both isopropyl and propyl and see which one works better. So prediction can tell you, and then you have to test it experimentally. If you only do prediction, it's not going to work. If you make a lot of compounds, that is very good, but computational modeling, after you make some compounds and after you get some data, then you can also do computer modeling to see whether using computer modeling is this telling you the same thing that you got experimentally. Then you know that your modeling is also correct. Computer modeling is developed based on experimental data, right? So the more experimental data you feed in, the more you know whether it is the accurate representation or not.
Like one more question. So I was just curious about the Gleevec. So you told that this molecule inhibits the ABL bond catalytic site. So
In the inactive form. I did not tell you that at that time, but Gleevec actually targets the inactive form of BCR-ABL.
like it doesn't interact, like it doesn't disrupt the interaction between BCR and ABL.
Nothing. So BCR is very far apart from the catalytic domain. If you remember the picture, BCR is on one end and catalytic domain is there, but because of the BCR, the way catalytic domain is inhibited, it is no longer inhibited. So it is in a different open conformation, but it still is inactive to a little bit. So what happens is that ABL is shut down in normal circumstances. When BCR-ABL is there, it is inactive. But Gleevec binds in such a way that it traps it in the inactive conformation. So this is something actually we were working on. So I was at Novartis at that time when this whole story was going on. So they had just discovered that Gleevec was, $1 billion drug for BCR-ABL at that time. But they could not figure out why, when you can inhibit BCR-ABL better in the cell versus when it is in the purified active form. So they even discussed with me, can you tell me why this is happening? So lot of people did lots and lots of experiments, and the end result was that this Gleevec inhibits BCR-ABL and locks it in the inactive form. So it can never become active. So that is why it is really active in the cells. Usually when you purify the protein, it is much easier to inhibit a purified protein than in the cell, but in Gleevec it is the other way around. That time it was not even known that kinases have this inactive versus active. The kinases have allosteric pockets. Nothing was known at that time. So Gleevec was very good in revealing the mechanism of actions of kinases also.
Like in normal cells, what is the function of ABL apart from cancer cell?
It is involved in a lot of signaling pathways. I have a friend who has spent like 10 years working on ABL. I don't know the specific signaling pathways, but it is involved in a lot of different cell pathways. It's a very good guy. See, the theme that is emerging, that the cells, if they're expressing something, these things are good for the cell. That's why they are expressing. Okay? But over time, if they become overactive, everything overactive or underactive is bad. Everything has to have the optimal activity. So if you work very hard, you also have to take a break. That is very important. Everything has to be in balance. We have to have time to regenerate. So anything too much good is also bad. Too much, CDK5 is activated, you have really good memory. But if you sustain that activation for too long, brain is going to degenerate, right? You smell good things, after a while you cannot smell anything because the system has to regenerate itself. So Abel does a lot of signaling pathways. I don't even know all the pathways of Abel. So don't think for a moment Abel is bad. If you remove C Abel, that's really bad because they all have function in the cells. But BCR Abel is not a regular protein. Some kind of really chromosomal translocation is occurring, which should not occur. And that gives rise to this really nasty protein. Abel, C Abel is a good guy. So, yeah. Yes.
Ma'am, if there is a family history of inheritary cancer, like genetic cancer, so can we perform the gene therapy of
You definitely
BRCA1
can.
BRCA2
Definitely.
in the zygotic for blastocyst stage, like of the offspring when two couples are in a marriage, so
You
when they
know,
want to have a child to reduce the chances of him developing a cancer.
you might have seen TV shows where they are genetically engineering the offspring. So the child is very smart. The child is not prone to diseases. The child is a superhuman, right? So these kind of concepts are not new, but there are a lot of ethical concerns about tampering with the embryo like that. Huh? Yeah, CRISPR also has a role on it. So anytime you touch the embryo at that stage and you start engineering it, imagine that most of the people are having normal children with normal intelligence. Think about a scenario, and some couple, they are very rich, so they play with the embryo and their own genes are not that great, but then they create a superhuman because they have the money to change the genes and everything. So if that is happening, that is going to, then the parents will start complaining that kid cannot be in school with our own kids because this is an artificially created kid, right? And there will be tons of ethical concerns there. So when you touch something at the embryonic stage to create that kind of thing, you have to cross a lot of ethical things. Some years ago, human fetal neurons were very common. So when somebody accidentally has miscarriage, or even when people get abortion, the embryo, their neurons are extremely useful in discovery for neurodegenerative diseases, right? So my friend who is an Alzheimer's disease scientist, he had access to that because he works in medical school. So he did several experiments for us in human fetal neurons. But then the ethical concern things arose and Congress voted against it. You cannot use embryo for this thing. They are actual life. Even if they are 2 days old, it is actual life, and you are killing embryos which cannot defend themselves and things like that. So all that research tanked. So basically, in principle, that can be done, okay? And eventually it will be done. I'm thinking you can fix the problem, and it is definitely possible to fix the problems at that stage. But having said that, people are still trying to develop therapies. Even a person is adult, that can be fixed. So these days what they do, that sometimes women, when they are relatively old and they want a child, so women who have their children when they are younger, their children are less likely to have diseases. But these days women in 40s are also giving birth to babies. But if the women are in their late 30s and approaching 40, or if they are in their early 40s, there is a very strong likelihood the children who will be born to these mothers who are relatively old in terms of childbearing age, these children can have Down syndrome. Okay, so this is at least in US, that this is a insurance approved thing, that these pregnant women have to test their embryos for Down syndrome. And Down syndrome is extremely bad disease, mental disease. So you don't want to bring a child with Down syndrome, because more than parents, it is the child who has to live that life with that. So in that case, if it is detected early, you have to find out. I don't know what is the policy, whether you can get it aborted if it is at early stage, or whether you cannot get it aborted. That I don't know the policy right now, because this is a very sensitive issue. But in some cases, it is allowed to test the embryo for really bad diseases. But I can assure you, sometime in future, you should be able to correct it at the embryonic stage. And in relatively near future, we can do BRCA1, BRCA2 fixing in adults. So we don't have to undergo surgery to remove organs. We can fix it. Muscular dystrophy, I'm really hoping you can do genetic therapy. Cystic fibrosis, you can do genetic therapy. Go ahead. I mean gene therapy, not genetic therapy.
So ma'am, about BRCA1 and BRCA2, they are maternal, so they travel maternally. So can they be treated in the ovary, the egg cells itself, can we...
You can. That's what I'm saying, that it can be treated, especially for in vitro fertilization. You can treat the things, but they are not done right now. When you are tampering at that stage, you have to get a lot of approval. So that is definitely doable.
Ma'am, because egg is not zygote yet, so that might be less unethical or problematic.
The thing is that the people who make the policy, they are not scientists, right? And everybody has their own view of that. So yeah, it can be done. It definitely can be done. But keep in mind, CRISPR-Cas9 is a very new technique. It did not exist 10 years ago. So now that we have more and more and more powerful techniques, we should be able to do these things. And you know what? I mean, this is my personal view. If we can do it, and we know BRCA1, BRCA2 causes this kind of horrendous life, I would change it at the X stage if I could. Because if you know something, you're not making a superhuman, you're not cheating in terms of intelligence, you are just giving a person a healthy life. And if you can, see, we all take medicines, right? And medicine is not bad if you can keep it. We all take supplements, it's not bad. If you can keep your body healthy, why not, right? We are not cheating by keeping ourselves healthy. We are helping ourselves, and we are helping everyone around us by being healthy, right? And we are contributing to society. So if somebody, for no fault of theirs, has the tendency, or they have inherited wrong genes, why not correct it? And I'm sure it will be approved, and it will become a common procedure. But keep in mind, the science is going on regardless of everything. You can always take eggs from some other organism and check it out. So science is not stopping because of that. And I know personally some really some outstanding scientists, like really the 0.01% of this world, who are just truly, they do all these things despite hurdles. They try really crazy ideas, because these crazy ideas one day will give you crazy good results, You guys are so tired. Go ahead.
Ma'am, this is one hypothetical thing. Like in elephant, we see there is a higher abundance of P53 protein. So,
Really?
yes, ma'am. So that's why they do not have any cancer.
Okay.
So why can't we replicate these things to humans by using a promoter enhancer kind of thing?
You know what? It is very funny, because I don't know. I did not have access to internet today. But I read this today, and I don't know how I read it today, because I did not have access to anything. So maybe I was reading my own lectures somewhere, because there is too much information in all these slides which I don't go through. But I read it this morning also, that too much of a tumor suppressor is also bad, because the cells will not even proliferate. So if you have too much P53, our cells will not even proliferate, and we need cell proliferation for survival, right? We share skin all the time, our stomach cells are proliferating all the time. So too much 53 is very bad for us. P53 is phosphorylated during cell division to switch it off. So you don't want too much 53. So keep in mind, nobody is perfect. You have to use everyone at the right time for the right amount of, like, right amount, right timing, then they need to disappear. So too much 53 is also bad, because no cell division can ever occur if there's too much 53. So we have to achieve the right balance. So think about it in another way now. Our physiology is so complicated, so many things. How do we actually achieve the right balance? You and me, right? We don't even understand what is going on in our body. How do we achieve the right balance? We have to do 2 things, 3 things, maybe 4 things. Not having too much stress. We have to learn that. Stress will happen. We have to learn how we respond to it, right? We can have a happy attitude about it. Or we can, there are always ways. You can go running, you can do something you like, right? Everyone has a different way, and we have to use different strategies to deal with it. We absolutely have control over our diet. That is something. And then no one in the world is that busy that they cannot exercise, right? We eat, we always have time to eat. We have to find some time. When you come to work, instead of taking the bus, I mean, of course not from Indore city, but for short periods you can walk. In US, they will run in the morning, they will jog in the morning, but when they go to the supermarket, they have to park the car. They will go round and round and round and round to park closest to the shop so that they don't have to walk even this much distance. But in the morning they are running, right? So you can park your car at a distance and you can walk. We always park our car in the first floor so that we don't have to take the stairs. You can park it on some other floor and you can take the stairs up and down. If your office is on the 4th floor, you can take the stairs to go up. I went to Japan, right? And I spent 3-4 weeks there. And in Japan, there is elevator in every building. Not even once any single person will take the elevator. Not even once. Every single time they will take the thing. I live in US. We are used to taking the elevator even to 2nd floor, right? So, and I was wearing high heels, and I never wear high heels. Never in my life. In Japan, I chose to wear high heels. I hated climbing the stairs. But when everyone is walking up and down, what would you do? When you go to subway, nobody takes the elevator. Everybody, every single person takes the stairs up and down. So my husband said, he's very active. So I had no choice but to go up and down with him all the time. 1 month later, I came and everybody said, you look so different. 1 month of exercise for a person who never did any exercise. It was such a life changer. That's when I realized how good it is for you, And it was very easy, something I could not dream of. I loved it at the end of my trip. It was so nice. Like, I would take stairs every single time, going up and down. And the place I was staying in the university, right? Metropolitan University in Tokyo. So what they have done, and only Japanese people will do that. So they have made a university at a very high elevation. So there are like hundreds of steps you have to climb. If you have to climb hundreds of steps, and then they have acres of land where the whole campus is there. Imagine, no car can ever go because it's hundreds of steps, and they have not made a road. So once you enter the university, you have no choice but to walk miles and miles to the place you are staying. So even if you're coming at 2 a.m. in the morning, you have to still walk to go to your place of residence. If you have to come out, you have to walk that much thing. So you are forced to walk all the time. No wonder. Your body is so fit at the end of the whole thing. And Japanese people are very, very fit people. And when they make the line, there is not even a single person who is out of this thing. In our country, The line has no relevance because there are people everywhere. You have to see the line there, right? And nobody takes. You come to US, every single person will take the elevator. In the morning, they will run, they will jog, and then they will not climb the stairs. They have to take the elevator. I am included in that, right? So my point is there. It is so complicated. We can still do exercise without understanding the physiology. Body will take care of it. You know, we eat healthy food, body will take care of the rest. You know, so the least we can do after we hear that is to really take care of ourselves. And trust me, it is like, you know, if you live a healthy life, it is true also that healthy people also get cancer, right? I don't know why they get cancer. It is pre-genetic disposition or something. But it still helps tremendously. You know, very, very, very few people will get cancer if they live a healthy life. That is definitely true. And, you know, cutting down on alcohol, never getting exposed to smoke. If you live in Delhi, you are exposed to pollution. I don't know what is the, you know, how you can protect yourself from that, but if you don't live in Delhi, at least you are protected from that kind of bad thing. And this is very serious, especially about Delhi, like the kind of pollution is in Delhi. It's very serious. I mean, somebody will figure it out someday, and but at least people are recognizing it is very serious. So.
But I have noticed that leukemia is more common compared to other cancers in kids and teenagers. So is it true, and is there any specific reason for that?
I know for sure that leukemia is more common in kids. That I know. I don't know why they are more common in kids. That I honestly don't know. Because I have a friend, he works on leukemia, and he was telling me the same thing, that you see these younger people with leukemia. I personally know a lot of people who have developed leukemia as childhood, in their childhood. I don't know the reason for that. Yeah, I really don't know that disease, why. It could have a genetic component to it, and it could be something is wrong, so the bone marrow is affected. I have no idea. Actually, I asked him the same question, that why it is so common, and I don't remember he gave me any clear answer. I would have remembered if it was known clearly. But leukemia is a very strange disease. Like some people do not survive, and some people live their life while undergoing treatment. Yeah, in some cases it can be fixed also. And there are many kinds of leukemia also. I told you about a student who had leukemia in my lab, right? And she is doing extremely well. And she was diagnosed with leukemia when she was 2 years old. So, but then she underwent some therapy in Singapore. She is from Singapore, and it is gone forever, like kind of thing. Like she doesn't, initially she had to get some kind of therapy every 3-4 months, serious therapy. She had to be hospitalized. She was out of, you know, like she could not come to work for 2-3 days while she was recovering. And then she got some treatment in Singapore, and now she does not have to do anything. So, yeah, I don't know much about exactly what is the reason and what kind of therapy or things like that. There's obviously bone marrow transplant, but that does not give you complete remission. Yeah. No other questions? All right, guys, we finished today early, 4 o'clock. Not bad. So I'll see you guys tomorrow.